Our next speaker is, as you know, Professor Ramamurthy mentioned that we have to integrate things, okay? Modern days problems are way, way more complicated. And sitting in silos, we can't handle those. And he rightly pointed out that we have to integrate lots of different approaches to handle these problems, the bigger problems. Following on that track, we have a very interesting speaker. She's Saroja. She's a scientist with expertise in genomics. She earned her PhD from the University of Vellore and has extensive experience in cloning, gene expression, microarray analysis, next generation sequencing. You would have heard this word so often these days, NGS. After many years of hands-on wet lab research, she now serves as general manager of a business at Genotypic Technologies. It's one of the finest company in the country which handles molecular analysis of various DNA, RNA, and metabolite samples. In this role, she supports clients in study design, experiment planning, and selecting appropriate NGS applications for the research. And additionally, she plays a key role in the adaptation and implementation of new technologies at Genotypic Technology. And it's like she works in one of the premier, establishment in the country, helping lots of academicians across the country, gearing up for these modern high throughput platforms. Saroja, please. And she has 2 sessions, okay? So I requested her to do one a broader theoretical session and then one like a practical session. So you can open your laptops and keep looking, and she will be helping you, okay? Going through with lots of various modern high throughput platforms. Saroja, please. Thank you so much for coming. And she's located at Bangalore, okay?
Thank you very much, Dr. Subhash, for your kind introduction and also for the introduction of our company, Genotypic Technology. So for us in genomics, we see everything as DNA, RNA, right? Not like insect, plant. Though it comes from those living organisms, end product what we see in our labs is crush everything, extract DNA, RNA, and look at the molecular level to understand what they are doing or what they are. So my talk today is also based on that, like how do you use DNA molecule essentially in identifying the organism and what are the different approaches and how it complements the taxonomical classification with molecular methods. A very brief introduction about our company. Since I represent a corporate, it's mandatory for me to just give you an introduction of our company, 2-3 slides, nothing more. So the company is now 26-year-old in the genomics area. Ours was the first genomic services company in India. Got many technologies first in India, including microarray and next generation sequencing technologies. We are also first official partners with nanopore biological pore-based sequencing technology in India, provide services on that platform. The latest addition to our technology platforms are PacBio Onso, which is very similar to Illumina-based sequencing technology, short read technology, but higher accuracies than Illumina technology. And single cell genomics, where people are looking at how changes, both epigenetic and genetic changes in the single cell level are contributing to biology. So that is our latest addition. We are about a team of 70 plus employees in the organization, out of which we have more than 12 scientists taking care of various workflows in the company and helping our clients in execution of their projects. This is broadly what we do. Anything to do with DNA and RNA, we will be able to support people. These are the technology platforms what we have, which has Onso, Nanopore, Agilent, 10x Genomics, and Somologic proteomics technology. And we do pretty much from all the applications of next generation sequencing, from whole genome sequencing, metagenome sequencing, transcriptomes, small RNA sequencing, chip sequencing, many other kind of sequencing services to cater to your needs. So now we will come to the topic of the day, or what is interest to all of us. Taxonomical classification of insects using molecular methods is what I'm going to talk. But just to give you a little background, as Dr. Ramurthy already mentioned, most of the taxonomical, or always the taxonomical classifications, are based on the morphological characteristics. You look at the insects in detail, understand and classify them. And it is also challenging due to very highly similar morphologies between the insects. And usually identification of the insects is based on its adult stage. Only when it is morphologically nicely differentiated, you will be able to identify its features and classify it. And also male genitalia. So I did, since it is highly dependent on morphological characterization, it is often time consuming. I'm sure it will need a lot of detailed workout to understand its morphology and classify. Needs expert taxonomists to pay attention to minutest of the details to differentiate between two closely related organisms, and also prone to misidentifying cryptic species which are very highly similar to the one which I'm showing an image here. The insect A has been classified quite some time ago. If I see the date, it's about 1800s. It has been explained its morphology and classified. Whatever organism you are seeing on to your right side has been identified very recently in 2009. If you see, both of them morphologically look very, very similar. So it's very difficult to identify and classify them only based on morphological characteristics. This is where the DNA barcoding technologies play a role. So we all know, right, what is barcode? Barcode is just a sequential line, dark and light, some thin, some thick. But this much information is, or scanning by this information, we get to know everything about the product. We usually use barcodes being used in supermarkets and shops, right? They just scan a barcode, they know what product it is, when it was manufactured, what are the contents, everything, pricing, everything, right? So similar to this barcode, the DNA barcodes are going to add value to taxonomical identification. Paul Herbert has been called as father of DNA barcoding. Was as late as, I think, in 2003, around that time he established importance of barcoding and barcoding for all different classes of organisms, different markers to be used for different classes of organisms. So barcodes, now we go to DNA barcoding. How it is done and what it is done. So barcoding is a technique that identifies a species, be it a plant, bacteria, animal, whatever it is, based on a very short DNA sequence coming from a specific gene. We will discuss about what are all the genes, what are all the regions in the later slides. But understand, it is a very short DNA sequence from the organism that is sequenced to identify what organism it is. It is a very powerful taxonomic tool because it will allow us to identify and segregate highly homologous or highly looking similar insect species. It is considered as an alternative tool for biodiversity identification in India as well as all over the world. It is a very well accepted methodology. What are the advantages of DNA barcoding? They are universal. Be it in India or anywhere in the abroad, they are going to use the same set of markers for identification, and between the organisms also same set of markers are going to be used. So the universality is one, it makes it very easy to apply everywhere. Highly reliable, because there is minimal expertise needed. You need to have some expertise in DNA extraction, amplification, and sequencing, but there are a lot of extensive databases available which will match your DNA sequence and tell you what organism it is. So that way the expertise required is minimal, and it is scalable because there are a lot of improvement in the sequencing technology. There are high throughput technologies. It is highly scalable. At a time, you can sequence hundreds of insects, barcode hundreds of insects in a very short time and identify what genus and species do they belong to. As I said, it is highly accurate, fast, and also cost effective because of the advanced technologies. So coming to DNA barcoding regions, there are different regions identified based on different class of organisms. In case of bacteria, the highly used barcoding region is a 16S ribosomal RNA region. Though I write it as a ribosomal RNA region, this is coming from DNA. rDNA or DNA locus that codes for ribosomal RNA is what is sequenced. So if you see the diagram there on top, the 16S ribosomal RNA is a longer sequence with few conserved regions and few variable regions. These conserved regions are conserved across different species of bacteria. They'll be highly homologous. So this is the region where the primers are usually designed so that it can anneal to those regions and amplify the variable region. But variable region are the ones which are informative to us. Those are the ones that are different between two different bacteria to identify that they belong to two different species. That is about 16S ribosomal RNA. All markers pretty much work on the same principle because they are evolutionarily derived. They will have a lot of sequence similarities among them, and also evolution plays some role in selection. So because of that, few SNPs or few bases are different. So those differences is what will help us to classify them as a different species. So for fungal identification, usually used region is ITS or even 18S marker. In case of animals, cytochrome C oxidase subunit 1, cytochrome B, 12S ribosomal RNA are used as markers, and all these above three markers are present on the mitochondrial DNA. They are not on the chromosomal DNA. And of many a times, 18S ribosomal RNA marker is also used, and that is present in the nuclear genome of an organism. In case of plants, MATK and RBCL genes are used as DNA barcoding genes, and both these are present on the chloroplast genome. So now you saw, right? There are three different markers and most of them are on mitochondria. Why mitochondria is so important, or why the mitochondrial genes are chosen for DNA barcoding in case of these insects and animal kingdom. Mitochondrial DNA markers play a major role in understanding ecology, evolutionary biology, and phylogenetics of a species. The main features that make mitochondrial genome or mitochondrial genes contributing to DNA barcoding is their high copy number. Per cell, if you see the genomic DNA copy number is 2 in the diploid organisms, whereas mitochondrial copy number vary, they are huge. So even in your sample is very little, through PCR, the available DNA, since the copy number is more, you will be able to amplify and see them. That gives it an advantage as a marker. Another one is maternal inheritance. So mitochondrial mitochondria is always inherited from a mother. So there is no recombination happening. So you get authenticated markers from there. Near neutrality means though there are mutations in mitochondria, usually they are not very lethal or detrimental to the genome. They are mildly, damaging kind of mutations. So that gives an advantage for its usage as a marker. Technical ease of use, lack of recombination, I already told you. And rapid evolutionary rate when compared to nuclear genomes. It is known to be almost 4 times higher evolutionary rate in the mitochondrial genome compared to nuclear genome, which makes us to identify new species very quickly. And also availability of extensive reference libraries for COI gene. If you see Barcode of Life, which is a database for markers, you see a lot of repository from the COI gene. So when you sequence it, you also have a good database to compare the gene. So that makes it advantageous. So there's some information about the insect marker genes and their lengths. Why I have presented it here, you would know in the next slide, but they are all have longer lengths, whereas marker region, which can differentiate between 2 species, are very short if you see. Some are only about 200 bases. The COI gene, which is widely used, is about 658 bases. And depending on the organisms, the length of the gene and the marker region would also vary. And there are 3 main types of barcoding. When we talk about barcoding, there can be a single locus barcoding, multi-locus barcoding, meta-barcoding. So I would touch upon all these 3 to tell you on the advantages and how it is used. So this is the overall workflow of DNA barcoding. What is the wet lab workflow, and then we will do some analysis workflow as well. It all starts with insect tissue. Whatever insect you want to identify, it will start with that tissue. So usually I see insects are either stored as frozen insects or in ethanol. They are stored in ethanol. So usually these 2 methods give very good quality of DNA. But it is also now a demand or the requirement that we don't kill all insects just to barcode them, right? Because you want to identify, you kill them, you mash them and barcode them. No, just find some methods which will enable you to still identify an insect without killing an insect. Those are the methods like you collect insect poop and try to barcode them, insect eggs rather than killing the insects. Because barcode is a signature. At whatever stage the insect is, the signature is going to remain the same. So try to, as much as possible, use non-invasive methods. When insects are available in abundance, you can definitely use direct insect for lysing and extracting DNA. So that is first aspect. Second aspect is extraction of DNA. Insects are also very have chitinase cell wall, different kind of structure. So because of which efficient lysis of the cell is very important, and after lysis of cell, retaining the genomic integrity is also very important to do the barcoding studies. Once the DNA extracted, we do a DNA QC on agarose gel to check their integrity. It need not be very high molecular weight, but it also plays an important role because in the previous slide I showed you some of the markers were longer, right? When the markers are longer, 1.5, 2 kb, if your DNA integrity is not great, you might not get a good amplification. So that's where it is important. And based on your integrity also, because there are several markers available, if your DNA integrity is bad, you make a decision what marker you want to use. You want to use COI for sure, because it has a better database, everything. But if it is not even amplifying, you miss the chance of at least getting some information out of it. Then you use a marker which will have shorter amplicon. Even with the degraded DNA, it would work. That is a third aspect in the workflow. It is not The direct aspect in the workflow, but very important aspect, is the primer design. There are DNA barcode regions, and as I told you already, there are regions that are common across all insects or majority of the insects, and there are variable regions. So depending on the insect group you are looking at, it's very important to generate consensus sequences of known insects in that class, and then design your primers to the conserved region. So it will have the ability to amplify maximum number of insects coming in that class. Mostly primers are universal. They do work well on broad insect species, but in some cases we have seen that they do not work. In those cases, we download sequences only from those species and arrive at the consensus sequence and then design primers to ensure these are amplified. Once the primers you will have in hand before, and then from the genomic DNA isolated, you do a PCR amplification. Polymerase chain reactions are the ones which will amplify your DNA to many folds. Because if you want to see them directly, it will be very low in copy number. We might not be able to see it. And it will also have other background. So PCR is the technology that gives us power to amplify or magnify the region we want to look at. And then the PCR product is sequenced on the sequencing technologies. Now, while using the sequencing technology also, depending on your requirement, you would choose a technology. So, Sanger sequencing is a very standard technology for DNA barcoding. When you have a single insect sample, pure insect sample, a single pure amplicon, give you a very nice sequence on Sanger sequencing, and you will be able to identify what organism it is. The other next generation sequencing technologies are used when you are using different multi-locus barcoding you are trying to do, or you are trying to sequence environmental DNA to identify insect species from that. So I have few case studies and references which I will present in the later stages. So once you generate sequence, you do analysis, do a phylogeny to see what is it very close to, and then if your insect is different or whatever, you submit it to the barcode library so that it becomes a part of library sequence which somebody else can use down the lane to refer, compare their sequences. So sometimes the single locus, I think that guy went off. He asked a question, right? How do you differentiate? Sometimes DNA barcoding is also not enough to differentiate. Like taxonomy, DNA barcoding also will have some limitations. How do you overcome those limitations? Sometimes single locus is not enough to resolve between two species, and this happens usually when the species separated by a very short divergence period. They can contain introgressed genes from closely related species, and sometimes the different species of this will have same haplotypes. For example, few spider species. So in those cases, it's very difficult to resolve between species using only one marker. Then, because there are multiple markers available, you can use a combination of different markers to identify the insect species. So you see the image there. You just like in the previous slide what I showed you, you do PCR amplification of different loci, but you pool them all together. You have an option of sequencing them individually and analyzing on Sanger sequencing, but that would be again time consuming. Because Sanger, you have to do forward, reverse for all the barcode regions. And if the barcode regions are very long, you will have to also have middle forward and middle reverse primers to cover the entire region. So in those cases, you make use of next generation sequencing technologies like nanopore, which will allow you to pool all the amplicons of an insect and sequence them and analyze them together so that you understand what species it is. If one barcode is not able to resolve it, another barcode, adding another barcode will definitely help in adding information and resolving it better. So this is from a paper where they have shown the utility of using multiple barcode sequences in differentiating a species. It's not that everything has a saturation, right? If I use 10 markers, I'll be able to differentiate better. No, it's not like that. Everything has a saturation. But definitely when one marker is not answering your question, using multiple markers would definitely add more information and we'll be able to differentiate. So we covered single loci barcoding. Second one was multiple loci barcoding. Third comes a meta barcoding. Meta barcoding or metagenome is a terminology used for environmental DNA sequencing. They are not essentially pure DNA coming from a single insect or single sample. They will be coming from a mixed species or mixed samples. For example, the insects captured in traps, they will have multiple insects. So rather than sequencing multiple insects individually, all their DNA can be pooled and sequenced to identify what are all the species present in that traps. So this is this kind of a workflow employs next generation sequencing technologies. So I was coming across few references where eDNA from soil, water are examined to see seasonal changes in the insect population. So even in those cases, there will be mixed population of insects present over there. Insect gut metagenome is done to understand what is the bacteria or fungal population colonizing insect, which might be contributing to its health and disease. Flower samples to understand what are the pollinators, and identification of rare and endangered species is also possible through eDNA sequencing. So as I already mentioned, metabarcoding uses next generation sequencing platforms. It's high throughput simultaneous sequencing of multiple organisms. You can either do amplicon sequencing, PCR amplicons you can make and sequence, or you can do the whole genome, metagenome kind of a sequencing to identify insects through other genes as well, not only marker genes. And nowadays there are a lot of portable sequencers technologies coming up. So that would help to establish this on site and do your barcoding quickly. So I'm just put together what are the different sequencing technologies available and what you choose when for barcoding. The most used barcoding technology is Sanger, because we were mostly looking at single gene sequencing. So it's a big dye terminator technology. You uses dideoxy NTPs, and the fragments are separated on gel and then imaged. Read lengths about 600 to 800 base pair can be read easily. When it is longer, you sequence from both ends. But when they are about 2 kb or something like that, you sequence even from the middle to ensure you are covering the entire length. The sequencing data format is Chroma, which is converted into FASTA format and used for analysis. I'll just show you in my next session how it is done. Whereas short read platforms are like Illumina platform. They have a read length limitation, but they can sequence up to 300 cross 2 as a longer region. So most of the barcodes which have lengths about 550 to 580 maximum can be sequenced on this platform, because there will be two reads, read 1 and read 2. If you get an overlap, you can stitch those reads and use it for your diversity analysis. And the file is in the FASTQ platform, FASTQ format. And the latest, the third generation sequencing technology is nanopore platform. It doesn't use any kind of imaging technology. It's a biological pores through which the DNA or amplicon is passed through and read. I have a just one image of how the nanopore sequences work. These are usable on field. Wherever, it doesn't need any fancy establishment, very simple DNA extraction and very simple PCR, basic PCR machines are enough. Then you sequence that on field. It's being used in Amazon forest, African countries, many areas for surveillance and identifying new species. So DNA passes through the pore, and when it passes through the pore, there is a change in the electrical signal, which is read as squiggles. And these squiggles, the machine is trained to convert these squiggles into bases, DNA bases, and that's how it is read and sequence is made sense of. Now coming back to technologies, to do a Sanger sequencing, you should, whenever you have a pure single insect sample, you can do barcoding using Sanger technology. If you want to use multi-locus sequencing, then you need next generation sequencing technologies. Sanger is doable, but it would take more time and effort. And in a single reaction, you can sequence multiple amplicons, and they also retain the entire sequence information. Actually, it's live base calling. As and when it is sequencing, it will align and tell you what organism it is. Yes, it will just initiation will take some time, but once the strands start passing through the pore, the mechanism is live base calling, and when it is base calling, you can do the blast. So then and there you can detect the organism, and it will match to NCBI database and tell you what organism it is. There is DNA extraction process. Yeah, so they have a very minimalistic setup which for DNA extraction is needed. And then there are sequencing adapters you add and then sequence. So PCR machine also some basic instrument comes, not very fancy ones which are usable on field. And nanopore, you can even plug it to laptop and use. Small battery backup is enough. Nanopore was not accurate when it came as a minion, but now the chemistries have improved. Now the latest R14 chemistry is accurate, more than 99% accuracy. And when we are talking about accuracy, it is also about the consensus sequence what we look at the end of the day. And those consensus are above Q20 quality. So that gives us more confidence in identifying the organism. So once we sequence and once we have the data, analysis of DNA barcoding data depends on the reference database, right? I have sequenced, but I have to see what is it close to. So this reference databases are very important. So BOLD is one such database, that is barcode of life database. And NCBI is a most comprehensive database. We use NCBI only because even whatever there in many other bases, it is always reflected in NCBI database. So that is the most trustworthy database. When we are doing a speciation or identifying it as a different species and placing it, we should always see that the variation within the species are smaller between the variation among the species. Intra should be lower than interspecies. And if a sample cannot be identified to the species level, especially in case of bacteria, they are assigned to operational taxonomic unit based on the homology or similarity of the sequences. And those are used in assigning the taxonomy. So these are some barcoding applications, DNA barcoding applications, which I have few case studies and examples how it is used in the field. One is, of course, biodiversity studies. Morphologically, for many of us, we will not be able to identify different species of insects. So it facilitates in vitro inventorying and monitoring of insect diversity, helps in assessing ecosystem health by checking changes in insect population, and it assists in documenting regional diversities of an insect. And also the DNA barcoding is unbiased, and since it's coming from the genome, it will remain same throughout its life stages. From larva to adult insect, its signature sequence is going to be same. So it will help us in identifying cryptic life stages. For example, most of the larvas would look quite similar. I do not know, I'm not a taxonomist, but Dr. Ramurthy, please tell me, that's what publications also say, that they look very similar. It will be very difficult to differentiate them during larva stages. But when you have a barcode sequence available, they can be identified at larval stages as well. And also at different developmental stages of insect, the barcode is not going to change. So it will contribute to identification of the organism accurately at any stage it is. Another application is to understand plant-insect interactions, pollinators and how they are pollinating, what plants are they, what insects are pollinating what plants, do they have any preference. So this is done through meta barcoding kind of a study, where you either collect insect samples and look at what plants they fed on, or you collect plant samples and identify what insect DNA you find in those plants. So these studies would also help in understanding insect dispersal and migration pattern. One such study we did at Genotypic, where a client was interested in dietary preference of bats, wild bats. These were insect-eating bats. So they gave us a fecal sample from bats. We isolated DNA from there, and then we amplified cytochrome oxidase 1 region from the fecal DNA isolated, amplified and identified what are all the insect species the bat was feeding on. So this will add information and value in identifying its diet preference, and also what are the insects present in that region. Another is pest and disease management. The very first thing, whenever you hear insect, is what we associate with pest. It is also important to manage pests to ensure that we are not incurring damages. So in those, these DNA barcoding plays a very important role. I have two case studies, or two studies to present. One is DNA barcoding done in Korea on vessels which come from foreign countries. So they actually sampled about 517 insect species, and they did DNA barcoding. In that, they could identify 145 species, insect species. Among them, 30 of them did not exist in Korea till then. They did not had any information repository of these insects existing in Korea, and among them 3 of them were identified as pest species. Now these are important, because if you do not do this, you allow or you make an entry, then you are introducing new pests in the new geographical region. So this way it will help in surveillance and identifying how the insect pests are spreading. The second case was is from Africa, where the sugarcane pest was identified. There were 4 different species of these insect pests, and among which particular one insect pest's larva was more detrimental to sugarcane plants compared to others. Since it is larva stage, it will be very difficult to identify, differentiate between them because they are all from the same genus but slightly different species. So DNA barcoding allowed them to identify these insects in the larval stage and take measures to control them from spreading. And it would also support targeted pest control strategies. Other examples are in conservation and policy. It aids in protecting endangered insect species by identifying them accurately. I came across one publication where it is in Switzerland. There are few insects that are indicative of freshwater quality indicators. And by monitoring by understanding what are all the species present in the water and how those are changing over the time would help you in monitoring the water quality indirectly as well. So this was one of the case study. Also from Indian journal, I got this DNA barcoding for identification of agriculturally important insect, which has used a lot of DNA barcoding technology to identify and classify them. So to conclude, DNA barcoding is a powerful tool for advancing biodiversity research because unbiased approach, it can identify from larval stage to adult insect stage. Robust methods from sample processing to analysis enable identification of species accurately. This is because we have methods developing every day. With very minute quantities, we will be able to extract DNA, do PCR amplification, and look at the barcoding. So this will, these methods will definitely help in better species identification. High throughput sequencing will help us in identifying large number of organisms in a short span of time. Integration of machine learning improves prediction and analysis. That's what most of the next generation sequencing technologies do now. We have pipelines, methodologies to analyze data and compare it with huge databases and do phylogeny. And the applications span taxonomy, ecology, pest management, and conservation, for which I discussed the examples. And continued innovation in barcoding technologies will further expand its impact. Today, one very important lesson I took from Dr. Ramurthy was integration. Taxonomy, biochemical tests, physiology, and barcoding. I don't think we can separate anything and see. This will be quicker to identify, but definitely they would complement each other in identifying and classifying organisms. Thank you.